animal and plant tissues. And we're done with that. So today we're going to continue on with how do cells replicate themselves, or how do cells divide. So we'll talk about the cell cycle and cell divisions. Okay. Now, of course, kanang natay naiduha ka ways in which cells are able to divide. No, it could either be I actually reproduce. Sorry. There are two ways in which organisms are able to reproduce, and that is through sexual and asexual means. When you say sexual means, you have a biological male and female gametes fusing together to form one organism that's sexual. But when you say asexual, the parent, not the parent, sorry, the offspring of the parent will be the exact copy of the parent. So basically, when you say asexual reproduction, this produces clones, no? Just clones. So there are organisms like that. Like, for example, your bacteria, kana sila, they are only able to replicate through mitosis, no, or binary fission. So asam sa magkuha sa ilahang DNA, so gikan na gyapon sa parent. And then the bigger usa naman yung parent, so basically it is a copy of his parent. So all of these, like for example, kung cholera nga bacteria, all of them will be the same. No, all of them will look the same. They'll have the same DNA, the look, everything. Okay, so that is asexual and sexual division. Not sorry, reproduction division. Nun. Okay, so let us start with the concept of DNA. So we talked about this already in the past, not last week, wherein we said that DNA basically gives your what's wrong with my voice? DNA actually um, translates your what is wrong with me? Hello? Ayan. Gisipon mo gud ko samo kayo. DNA actually dictates your hereditary nga kanang information, no? So it passes from, of course, from parent to child, so on and so forth. Uh, DNA basically, which is deoxyribonucleic acid, has the following. No, it has a sugar. That's a sugar backbone. Or sugar phosphate backbone. Nitrogenous bases, there are actually four, and they pair up, no? Ka-pair na sila. And then your notable ang shapes ng DNA because it is double helix, no? That's the shape of DNA. If you cut it in half using your DNA helicase, that will become your RNA. Half na nila siya. Okay, so these nitrogenous bases will pair up, no? Your thymine will bond with your not guanine, adenine, and then your guanine will bond with your cytosine. So that's Ta, and then G and C. For RNA, that is a different story, no? Diba, ang DNA, hereditary traits, no? It talks about hereditary traits. Your RNA, it has something to do with your proteins, or it will synthesize your proteins through the use of your ribosomes. Your RNA, on the other hand, will oh my god, my sipon. Will use instead of thymine, it doesn't have thymine, it has uracil, which will pair up with your adenine, and guanine pairing up with your cytosine. No? So very important among DNA because it stores genetic information. It tells us all of the instructions of our life, from our way of cell division to the physical characteristics that we have to our to our susceptibility for certain diseases, so on and so forth. So DNA is very, very vital, and it's very, very important. Ano? I can see from my window, no, nga kuan siya, overcast di karon no, dagum. Dagum ra dapit sa inyo. Mula na sad?
Yes,
Mula
ma'am.
na
Ulanan man diri.
kay natingala magud ko kay murag a few minutes ago. Grabe kaayo ka sunny, like sunny. Then karon ay dagum. Anyway, so um but March naman can you believe today's the last day of February? Dali ara kayo no, dali na kaayo
La.
good. Yes. Tapos na ang February. Dali na kaayo ang mga pangyayari. So, first day of March na tomorrow. So, um, But why is it that the weather is like it's like it's not summer yet? Mm-mm.
I miss you.
Anyways, madali pa summer, no? Like kanang you know it's summer because it's always like very sunny and very hot, but karon kasi ulan. Weird. Anyways, so as I've said sa una, DNA, imagine DNA is a very thin, long strand of thread, no? About 2 meters long and very, very thin. For it to be contained inside your cell, it needs to coil. Coil, as in like, was it bisa as a coil? Coil. Lu- Ilubad? Sakto ba ko? Ilubad? Ligid? No, ligid is different. Ano ba itong ano ni mo? Basta, kana siya. Okay. So, you're going to
Bugkos.
coil it. Ha? Unsa lang ga?
Ibugkos, ma'am.
Dili siya bugkos. Mura siya ituyok-tuyok ba? Lambod, that's the word. Lubad akong una-una jud the word. Basta something that starts with letter L. Thank you very much, Algie. So, lambod nimo. So, unsa ni paglangbod? So, kini siya, ang kini mong DNA strand, ginalambod ni siya using your histones, no? That's the protein. It's like a circle, then dara din mo siya ilambod-lambod ang imong DNA strand, which is the thread, no? Lambod mo ang imong tanod. Now, kini siya, when it is coiled, or kanang lightly coiled lang, it is called a chromatin, no? So kanang cell nimo nga wala na divide, like ano siya, static lang yung cell, kanang DNA nimo that is found in your nucleus, it is in the chromatin format. Daghan man ako format si DNA. Ang first niyang format kay kanang DNA lang din siya like tanod, ana long double helix nga tanod. And then pag i-coil din na siya, mo na siyang coil sa histones, mo na siyang gitawag na chromatin, no? So kanang imong static na cell, the DNA that is found inside of its nucleus is in chromatin format. Now, if imo siyang i-super coil, okay? So, na-coil na siya, no, kailangan pa man yun siya maggamay. Like, it needs to take up a smaller space. So, mo nang highly coil or super coil nimo siya. Pag i-super coil nimo siya, mo na siyang gitawag nga chromosome. But this chromosome, this will only be seen during your cell division. Dara na sa cell division yung makita. Kay, if pag human sa cell division manggod, mapurma na ang cell nimo. So, bali, from one cell, pag human sa cell division, may mo na siyang duha ka buok, ana, mag-static na din na siya, mag-remain na sila as just cells for the next couple of days, dili sila mag-divide. Kung if that's the case, kanang imuhang chromosome, which is highly coiled or super coiled, it will uncoil itself. I-loosen niya itself. Oy, it's time to relax. Ano siya? Time to relax. And mo nang ang chromosome dayon, mabalik na din na siya as chromatin inside of your static cell. So that's the difference between chromatin and chromosome. They're basically just DNA, however, their formats are not the same. Now, kita yung mga tao, no, we have a total of 46 chromosomes. Bale, 23 kabuka pares. 23 kabuka pares nga naman, nga nung pair up man sila. It's because one comes from your father and one comes from your mother. Now, why do they pair up? It's because both of these chromosomes, this one is a chromosome, this one is a chromosome, both of these chromosomes will be expressing the same gene. Like, for example, both of these chromosomes will be talking about, they will be expressing eye color, hair color, skin color, etc. Kana inana. So both of these chromosomes will express the same gene. However, they will have different alleles, no? Alleles. What do you mean by alleles? What is an allele? It is like a characteristic. It's like the value of that gene. Like for example, si father kay, let's say for example, imong papa kay si Chris Evans, no? Chris Evans has blue eyes. So iyang mata kay blue. Then imong mother, kasi inyong mother be, Si Song Ye Kyo, anak mo na inyong mother si Song Ye Kyo. Song Ye Kyo has brown eyes, so blue. Then ang usa dayon kay brown, gikan kang mother, no? So naay duha ka possibilities kung unsay ma-express nga eye color sa based on that gene, no? Based on those chromosomes. So in that case, it depends sa dayon. Nala na dayon mo come into contact ang imuhang genetics na dayon, which will be a topic. I think we will not be able to cover that karong senior high, but maybe in college you'll be able to talk about genetics na dayon. I know that you know genetics, so you know katong the fundamentals, that is like for example katong Punnett, the use of Punnett squares, which will give you a sort of like a bird's eye view on how dominance and recessiveness is being expressed, but it's more complicated than that. Of course, that's the one. That's the old, not old, but that's the most fundamental, but most elementary. But if you go to like epigenetics and hayon, dada yun mo makahilak. Okay. So, inana siya, no? There are a total of 23 pairs. The 23rd pair, the 22 pairs would be autosomal chromosomes, meaning they express like a lot of genes, no? Except for the sets. Because the last pair, the 23rd pair, this one right here, expresses your sex, no? Um, your X gene will be carried by your mother, biological mother, and your Y gene will be carried by your Chris Evans, your father, no? So, if your 23rd pair expression is XX, actually, sorry, your father also carries the X. Sorry. Okay, your father will have a lot of sperms, but sperm cells, but it varies, no? Na ibang sperm cells ng X, na ibang sperm cells na Y. Your mother on the other hand, ang egg cell na dyan niya is X. So if XX ang expression sa imuhang 23rd pair, what gender, not gender, sorry, what sex would that be? Is it male or female?
Female.
Female.
Female, ma'am.
Female.
Female. Good, good. Kadjut lang hapa, sa water. Just kidding. Okay, correct. This is female. If it's XY, that is
Male.
obviously male.
Male.
Ayan. But actually, naay ka ng, we will not be able to talk about this, sad ka, but naasa yung mga kanang wrong, kanabitong na-excess, like XXY and XYY, na yung mga, na yung mga mutations, you'll find out sa genetics. But, na yung mga sakit what do you call this? Hermaphrodites, no? Could come from these faulty expressions. So then, okay. So basically, it just says through that expression, no, it just says that your father's sperm cells are really influential. Sila mag-dictate kung sa gender ni mo, ay sa sex ni mo, no? Imong papa dyan, no? So if you notice in your family nga, bakaligay may babae, that would mean that your father's sperm count, not sperm count, but your father's sperm population, a lot of them express the X gene. Yeah. If you notice nga, dakan mo ka ng lalaki sa pamilya, your father's sperm population would heavily be Y gene coded. If varied mo, there's a very good mix of X and Y's in his sperm population. That's good, right? But naman ba nga we would prefer nga all girls or all boys, no? So it's like that. But in our family, in my family, varied mean I have two brothers, and there were supposed to be like two girls, but my sister died, no? So kuan kay me kanang perfectly balanced, perfectly splendid. Kita mo na nga salida sa kuan sa Netflix. So kita ito na The Haunting of Bly Manor. Splendid. Tanaw mo na, it's good. Huy, mag-uwas na mata kong Titan later. I'm so excited. Okay. Wala lang, random lang. Okay, so of course, we'll not talk about that. Let's go here sa cell cycle na mismo. Now, your cell cycle, not all of your cells are going to divide per me, no? Na may times na imuhang cell would just be static. You know, it's in stasis. It's gonna just exist as a cell. There would also be times wherein your cell will divide. It depends, no, because different cells in your bodies have different timelines, or they have different um, spans of time for them to live. Oh, so, like for example, your red blood cells, they could survive up to 90 days. No, after that, it will just die. Mo na na siya. So it will, so imong body sad, mag-start to sad na siya og cell division. Ako microphone si kahulog. Okay. So, for example, if you get wounded, masamad ka, of course, ang imuhang body kay, uy, mag-cell divide ta, paspas kay, kailangan na ito ma-heal si madam. Dapat ma-heal si madam. Respawn, na. So, kailangan sad mag double time sad si kuanso silang skin cells no to divide, no, so it depends, but there are also other cells wherein they don't divide any longer, no, like for example your nerve cells, kana sila, your nerve cells that are found in your brain, cells that are found in your spinal cord and your nerves overall, these these nerve cells, they don't replicate anymore. So once you have that certain number of nerve cells, that's it. That's it for life. If it dies, if papareho mo ni Professor X sa X-Men no nga, pak na pusilan sa kuan sa lumbar area. Wala na siya, na matay na iyang kana nga part sa iyang spinal cord. So mo tong na paralyze na siya forever. No, that's very sad. Nasad na noon ko, nakadumdum ko ni Professor X. So, in your cell cycle, you actually have three phases wherein your cell will be preparing for the M phase. No, this is the cell division phase. So, mas taas ang span of time. If you're gonna look at this, G1 up to G2, no, mas taas ang span of time in which your cell will prepare itself for the cell division, and then your cell division mismo is just really short. If you're going to count this out in hours, no, ang imong G1 to G2 nga phase, it will last for about 24 hours. About 24 hours. It's not really constant. It depends. It really depends. And then your cell division, may it be mitosis or meiosis, would be around an hour. Not siya ka-short. Really short. So, yeah, let's look into it. The stages of the cell cycle. Sorry kaayo ako ang sipon kay it has turned into phlegm. Sorry about that. Okay, so there are the three stages of the cell cycle. You have your interphase, na adiri imong G1, S, and then your G2 stages. So gap 1, synthesis, and gap 2, okay? And then your M phase would either be mitosis or meiosis. This is basically cell division, no? Asa dapat mag-divide imong cell. What's the difference between mitosis and meiosis, gani? Anybody? What's the difference in mitosis and meiosis? One ma'am ka lang. Ang usa ma'am kay ang sunod niya kay letter I, ang sunod sa M kay I, and ang sunod sa M kay E. Anybody who would be brave enough to tell us what's the difference between mitosis and meiosis? Yes, Arnold.
Ang mitosis, ma'am, kay involves a single round of cell division, and a meiosis dayon kay 2 rounds of cell division ug mo-produce og 4 non-identical haploid daughter cells, ma'am.
Yes, like in a nutshell, correct na, no? The mitosis would only go through the whole process once. Meiosis, on the other hand, twice. And they have differing number of daughter cells. But essentially, sakto si Regine, mitosis is a cell division of body cells or somatic cells. Body cells or somatic cells are those cells that are not your sex cells, basically. All the other cells in your body, like liver cells, skin cells, intestine cells, nerve cells, etc., etc. Meiosis, on the other hand, is a cell division of choice sa imuhang sex cells, exclusively for them. If your mitosis produces diploid cells, ay sorry, wait lang ha. Let me erase that. Your mitosis, wait lang. Your mitosis produces 2N, or what we call as diploid cells, meaning they will have 46 chromosomes, no? Your sex cells, on the other hand, this will produce haploid cells represented by a small letter N, and then haploid. And then what does that mean? Meaning half lungs up to 46. So each cell would have 23 chromosomes. So in a nutshell, that's the difference between the two. All right. Thank you very much, Ryan and Regine, for your answers. And then you have your last stage, no, or the last stage of cell cycle, that is your cytokinesis, which directly follows your telophase. And cytokinesis is basically the division of labor, but division of your cytoplasm. Okay, but the question is, what starts your cell division? Kasi mag-ingon nga, ay, mag-start at divide. Hoy, hold up ni. Charot. Hoy, hold up ni. Pag-divide na si mo cell. Dili ko kaawat sa social nga accent. I apologize. Okay?
Amazing.
So, ha? Sorry, Jean. Abi mong 5,000. Abi mong kwarta. Alright. So, actually, naay molecules or naay ka ng certain chemicals in our body that tells us that, oh, we need to start dividing our cells, no? There are regulators, no? There are two types of regulators. You have your internal regulators and your external regulators. When you're talking about internal regulators, you're talking about regulators that are found inside the cell. So, naa siya sa sulod sa cell. So, ang cell jud mismo nimo, naa siya proteins, okay? These are proteins that tells the cell that it's time to divide. Ano siya? If you remember, Katong, when we talked about the cell parts and function, I told you that there are free ribosomes that are floating in your cytoplasm. These free ribosomes actually synthesize proteins that are needed inside your cell. So one of the or two of these proteins actually ga-govern sa imuhang or ga-regulate sa imuhang cell division, no? And that is your cyclin, that's one. And then your protein kinase, that's the second one. These proteins actually regulate your cell division inside of your cell. And then, naapadod na siya hari-hari, no? Mo na siya ang gitawag na external regulator na ay mag-start jud ana or na ay mag-trigger ana. So, ang external regulator, that is a regulator that is found outside of the cell, and for this matter, it would be your growth hormone, no? Growth hormone as in like the hormone that tells you how tall you're gonna go. Like, ang growth hormone, kung daghan ka growth hormone, taas ang cake ka nga pagka-person. If masubraan mo, kang giant, kang Hagrid, na. Or if gamay na ka growth hormone, you will exhibit, like gamay na kaayo, no? You will exhibit dwarfism, nana. Or if gamay lang, di man jud abnormally gamay, but kind of low, your height will be like 4'9", 4'11". Dakan yung mga Pilipino ngayon na, raise your hand if you're 4'11". Bitaw, joke lang. Okay, so these are your internal and external regulators. They regulate your cell division, and it tells your cells when to start dividing. That's it. Okay, so here are the phases of the cell cycle. No, so we'll look more sa we'll look first at the interface. No, sa interface there are three stages. You have G1, your DNA, and your S phase, and then your G2. S stage. Anong S phase mo ngayon? S stage and then your G2. When you say G1 and G2, that's gap, no? Gap 1 or gap 2. Energy gap. I'm just kidding. Okay. Sorry. So, your ang pasabutan ni gap 1 o gap 2, just ano na siya? Just associate letter G with growth, no? Growth. Kailangan mo growth because you're preparing your cell to divide. Wala na siya. So, kailangan mo grow sa imong cell. How come? Unsa nga growth, ma'am? Growth in numbers. Like, for example, di ba na organelles inside your cell? Okay? So, basically, ang sa G1, tinidapit, there will be an increased number of organelles. Kay dili pwede nga mag-divide ka sa mo cell pero mahati sa dim organelle. Ang mitochondria kay hiwaon nimo ta half dili. So there will be replication sa dinhi, no? So madubli imuhang mga organelles, dubli ka number of endoplasmic reticulum, dubli sa imuhang Golgi bodies, dubli ang imuhang mitochondria. Ina na siya. So there will be an increased number of organelles and there will be an increase na metabolic roles. Okay, that's the, in a nutshell, metabolic roles. But actually, increased number of proteins, increased number of carbs, increased number of basically all molecules inside the cell. That's what happens in GAP1 or G1 stage. Now, in the S stage, this is where DNA replication happens, no? So, dinhi makita nimo imong chromatin. Very, very obvious diri imong chromatin. Mag-start na siya replicate. Replicate. So, basically, magdubli sad na iyang number, no? In number. So, if mo ni imong chromatin, it will then start producing, that's the first chromatin, it will start producing another one by DJ Khaled, but identical to itself, no? So, using the DNA helicase, iyahan nang i-splice imuhang DNA, ang imuhang kuan, double helix, iyan ang ihati, then mag-start din na siya replicate. So, katong mga nitrogenous bases, mag-pair up na siya with another nitrogenous base, and then nanay siya. So, bali, nanay duha ka copies na yun, all in all. So, I hope you still remember DNA replication, translation, transcription sa junior high. We will not talk about that now because it's not part, it is part of our lesson, but it's not the main focus, no? We will just talk about the stages. So that is the synthesis stage. S stage is synthesis. No, so good. Synthesis. Okay. And then, lastly, sa imong G2, which is your gap 2 or your growth 2 na stage, this is more focused on the preparation for mitosis. There's still growth. No? Dako pa din siya. There's still growth. Madako siya in size. Okay? But also, mas obvious dili ang mga structures that or prepare na o dubli ang mga structures that are used for specifically for your mitotic phase or your meiotic phase. It depends kung mitosis or meiosis na siya. Like for example, your spindle fibers, mitotic spindle that is from your centrioles, or they are derived from your microtubules and microfilaments, kana sila, no? They are made more obvious in this part, sa imong gap 2. So, in a nutshell, maura to ang imong phases of the cell cycle. Now, let's go individually per stage, no? Let's start with the G1 phase. Of course, na ay growth of the cytoplasm. And again, as I said kanina, magdubli ang number of our organelles. And in this part also, ma-produce nimo, madubli imuhang nutrients, proteins, katong gipang ingon ganina, enzymes, carbs, lipids, kung sa pa'y kailangan sa cell. Okay, mana siya. And then, this is also where your highest rate of protein synthesis is most seen, no? Kay, again, kailangan niya madubli tanan. Mora jud na iyang essence na magdubli tanan ang mga kinahanglan sa imong cell. And in this last part, the movement of centrioles of the centrosomes away to await and assist the events of mitosis. Okay. So, kani siya nahinay na imong centrioles nga move, no? Mag-move siya to sort of like the pulse of the cell. Hinahinay na siya. So kanay imong centrioles very important because those are derived from your microtubules and microfilaments, and they will enable to hold on to your chromosomes while your cell is dividing. Kaya kung dili sa hold on, if dili strong imong centrioles, dili sakto ang pag-divide sa imong chromosomes. Therefore, one cell could have like an extra chromosome and the other would be lacking. So, nanay something wrong na dayon sa cell. Okay. Now, in the in the S stage, which is the synthesis stage, this is where DNA replication happens. Okay, magdubli ang number of DNA. So, you can just read through this. This is in your PDF, but basically, mag-break ang imuhang bonds sa imong nitrogenous base, and then, mag-pair up dayon na siya to make 2 copies of the same DNA. So in the G2 stage, no, there is continued growth and the production of materials that are necessary for cell division. It's just like the same sa imong G1. Continue lang tayo sa ating mga tasks, ana siya. So protein synthesis happens here, but not at the same rate as that of found in G1, no? Dili na needed because G1 has already done that. So sa G2, igo na lang siya mag-prepare for the M phase. Okay. So in the M phase, of course, there is cell division, no? It could either be mitosis or meiosis. When you say mitosis, it is a cell division of your somatic cells, your body cells, that is. So basically, it only undergoes the whole process once, no? And it will produce 2 daughter cells. Meiosis, on the other hand, is a cell division for your sex cells, no? It starts off with a diploid cell, and then it goes through cell division twice. It goes through the whole process twice. Therefore, ang ihading products would be 4 daughter cells, and each of them are haploid, meaning they only contain half of the 46 chromosomes for human beings. So throughout the whole discussion now, we will only focus on human beings. So hindi na topic lang ng ubang animals because that will be quite confusing. No? So we will focus on the 46 chromosomes. And di na topic lang ng ubang animals because sometimes we will get confused because there are some animals that have, not some animals, but other organisms would have different number of chromosomes. And then we have cytokinesis. Cytokinesis basically is the division of your your cytoplasm. Now, sa animal cells, ang makita nimo sa pag-divide would be a cleavage furrow, no? Na ay kanang pag-divide sa cell kay murag muana din siya shrink, murag maginani din yung shape, then inay-inay din siya mag-go apart from each other. So that is a cleavage furrow, no? Meron ang cell membrane kining close siya, and then go away siya sa each other. For plant cells, because they can't do that because they have cell walls, no? Instead of a cleavage furrow, you will see a cell plate. So remember, katubitong drawing bito ni Robert Hooke bitaw on cells. Katong, di ba siya ang first siya ang naka-coin sa term, and then siya sa first nag-draw og ayo, no? If you remember, inana ang iyahang drawing sa iyahang nakita under the microscope. So, because your plant cells, they're rigid, no? They're quite rigid because they have cell walls, no? Made of cellulose. So, in that case, ang cell membrane dili mag cleavage furrow for the plant cells. Instead, it will form cell plates. All right. Now, of course, naasay mga checkpoints, no? Dili man pwede, the thing about your cell division, your cells all in all, they're quite smart because they can actually detect if there is something wrong during the whole cell division. Now, for example, if they can detect that there's something wrong during the DNA replication, like there are mutations that are happening na kanang dili normal, no? Mutations can be normal. Actually, mutations are normal. Like for example, your red hair. We don't have that here in the Philippines, but red hair kanang pareho kang Merida sa Brave, or kanang imuhang auburn hair, kana siya the cross between red hair and brown hair or black hair. Kana siya, those are mutations and they are practically harmless mutations. So if your cell can detect mutations that are practically harmful, na medyo harmful siya for the rest of the cells, it can actually stop the cell division from happening. So there are these checkpoints, no? There are 3 checkpoints throughout the whole cell division. You have your G1 to S checkpoint, or you can just write it down as G1. Pwede na siya. And then nakay G2 to M, or just G2, okay, that's na siya, na checkpoint. And then you have your metaphase checkpoint. This happens katong sa imuhang mitosis to it's PMAT, right? So the metaphase checkpoint will be found here in the second phase of your cell division or your mitosis. All right, so let's look at the checkpoints. So G1 to S checkpoint, it asks the following questions. Is there any damage in the cell's DNA? Just like what I said, there may be harmful mutations that are already kanang na-happen na siya before, pero wala na detect, so nag-continue og cell divide ang previous na cell. Then karon kay kung ano naman, you're much stronger, or your cell has detected it. Uy, harmful DNA mutation, so we have to stop this. Okay, so if there is any damage to the cell's DNA, then it will stop. It will enable your cell to diba G1, S, and G2. So if that will happen, nga na something wrong, ang cell na previously as a G1 phase, it will go to G0 phase. Okay, what do you mean by G0? Static lang imong cell. Like, nana siya exist lang siya din ha, pero it does not undergo mitosis, or it does not undergo division. So that's the first question. And the second question is a cell large enough with all the necessary energy reserves and doubled organelles? Because if there are no doubled organelles, it will not start to divide. Imagine na imong cell, no? Dili siya magdubli, unya mo push through pa jud ang division. So naayo sa ka cell na ang lugi ang usa ka cell, magkulang siya sa iyahang organelles. So that should not be the case. So that is the first checkpoint. The G2 checkpoint, or the second checkpoint, answers the following questions. Are the environmental conditions favorable for cell division to take place? Because cell division needs to have optimal nga kanang internal environment for it to happen, no? And if we are unhealthy, like we don't have or we don't reach, like for example, we don't reach the right range of internal or core body temperature, or dili sakto imong blood sugar levels, dili sakto tanan, or for example imong hormones, no, imong growth hormone wala siya nag-release or gamay ra ang gi-release niya. If it's not enough or if it doesn't meet the expectations of your cell, then the division does not happen. Okay. So dapat tanan things inside your body should be in a normal state. That is why if namay sakit, if you're gonna have a fever, like nana lugar, mag ano, hinay sad ay mong rate of cell division. So, hinay sad ka mag-heal, no? So, if you are sick, hinay sad ka mag-heal. So, that's the case there. Mo na siya ang what you call this, mo na siya ang i-meet na requirement, minimum requirement ni G2 checkpoint. The second is the DNA properly replicated? Because gikan naman siyang S stage, right? It's from the S stage. And then sa S stage, nag-replicate man siya sa imong DNA. So, if it is properly replicated, okay, push through. Ay, okay. Ano siya? Push through ta sa division. Pero if dili, because of a lot of factors, no, it could be factors in the internal environment. There might be something wrong sa internal environment nimo sa imong lawas. Or, naabay ionizing radiation, meaning ning buto ang usa ka nuclear plant, and then duol kay ka or you work there, so you were exposed to a lot of ionizing radiation, pwede na siya. Or you are exposed to x-rays a lot of time, pwede na siya, no? There will be repercussions to it sa pag replicate sa mong DNA. Or because of chemotherapy, pwede na siya, no? There are drugs that are strong enough to penetrate your cell membrane and your nuclear membrane, and thus your DNA as well. And then third is there a presence of DNA damage? Just like what I said ganina, your DNA can be damaged through a number of ways, no? Ionizing radiation, that could be one. Chemotherapy, that could be second. Or it could be conditions in your internal environment, or it could be, kamalian sa pag-replicate sa imuhang DNA. So those are the cases. So if that's the case, if these were detected in your G2 phase, G2 stage, cancel na. Cancel na to ang television kay dili happy, I'm not happy with the conditions. Ano siya. So, kini imong checkpoints, they are majorly quality assurance din na siya. And then lastly, we have your metaphase checkpoint. Now, in metaphase, ang imo remember, ani, would be M is for middle. Why don't you just meet me in the middle? Ano? So, this is the middle, wherein kini siya nga part sa imong cell division. This is where you can see your chromosomes, kana sila, connected to your mitotic spindle, there, chuk, 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 and they grapple onto your kinetic cores that are found in your chromosomes right there. And they have to be aligned right in the middle. Ano siya? Formation! Para sa yan! Ano siya? So, mo form na sa tunga, and then dapat mauna ang i-meet na requirement ni metaphase, no? So, ang question sa M checkpoint is, are the spindles properly attached? Okay, meaning, are the spindles, kini mga imuhang mga microtubules, no? Are they attached sa kinetic cores sa imuhang chromosomes, no? Because they function like this, they're like grapples, no? Kay in the next manggod nga phase, the anaphase, ilahang i-pull ang imuhang chromosome towards the opposite poles of the cell. So, inana siya. So, dapat naka-grapple or naka-attach na siya securely. Kay again, if dili siya attached securely and the division will occur or the division will follow after that or anaphase follows, naay usa ka cell nga lugi. Kulangan siya chromosome. Moo. Nasa usa ka cell nga, subraan na sad siya. So, that is already mag-mutate na imong cell. No? There will already be something wrong with the cell. So those are, or that is the main question of your M checkpoint. So kanimong mga checkpoints, they are just quality assurance ng mga checkpoints, no? It will guarantee that your body, kay, will be in its most optimum state. Charot! Sige. All right, so let us check your understanding. Identify the cell cycle phase or checkpoint being referred to in the following statements. All right, let's start with number one. This is when DNA replication occurs. What stage? Yes, Regine.
Interphase synthesis, ma'am.
Okay. Juniel?
S phase po, ma'am.
S phase. Okay. Any other answers sa kuan? Sa chat box? Synthesis, S stage,
phase.
interface. Yes, okay, that's correct. So this is the S stage of your interface. S stage. All right. Because again, S stands for synthesis, meaning there's a, when you say synthesis, there is this production of the second copy of your DNA, S stage. Second one, this is the first stage of the interface,
Gap 1, ma'am.
gap one.
Drop 1, ma'am.
Okay, G1, gap one, any of those. Okay, so that's G1. All right, number three. This refers to the division that occurs, division that occurs in somatic cells.
Mitosis.
Okay.
Mitosis po, ma'am.
Mitosis, my knees, my shoulder, my head. Bitoy. Mitosis, correct. Ayan. Number four. This checks if the chromosomes are properly duplicated. Sakto
G2 checkpoint, ma'am.
G2.
unsa?
G2 checkpoint.
G2. All right. Okay, that's correct. So G2 checkpoint. I-check niya kung sakto ang pag-replicate sa mong DNA, which happened in the S phase. So right after the S phase, si G2 siya na niya, sakto ba? The interpretation is correct. Yes. But it's wrong. Charot. Wala siya. Okay, number five. This refers to the division of the cytoplasm. What is this?
Cytokinesis, ma'am.
Cytokinesis.
Bungan sila bay. Cytokinesis. All right. Thank you very much for your answers. Thank you very much for those also answering in the chat box. Your participation is very, very highly appreciated. I was looking for the word in my brain. What word am I looking for? Okay, so dito sa kanang analogy, no? The first two words are separated by a colon. These two words are related to each other. Analyze this relationship in order to fill in the missing word that will complete the second pair. Okay. Number one, if mitosis is to nucleus, because basically ang imong cell division, imong mitosis and meiosis is the division of your nucleus, no? Cytokinesis, on the other hand, is a division of what?
Cytoplasm, ma'am.
Cytoplasm po, ma'am.
Cytoplasm. Naimanghod nga. Ay!
Ano ba yun?
Nag-apil-apil.
Manghod. Pil-apil. Number two, if plants is to cell plate, this talks about your cytokinesis there. Then animals is to? This is in cytokinesis. What is found for animal cells? Okay. I can see nanay nag-answer sa chat box. Cleavage, what? Cleavage.
Cleavage furrow, ma'am.
Furrow. All right. Great. Okay, great. Okay. Number three, if interface, you can see DNA in the form of chromatin. What about in mitosis? Ano? What do you call that? Kani siya nga form. Basically, it's just the hypercoiled nga chromatin.
Chromosomes, ma'am.
Chromosomes. Ayan. So, chromosomes. So, ang chromatin nimo, it is most apparent in the S stage, no? Dana siya mapangka jud nimo ang pagkapurma sa imong DNA as a chromatin. Remember, ang DNA, think of it like a very thin, long strand of thread, no? And it needs to be coiled, and it is coiled around a protein called histone. Now, when it is coiled, it will then become chromatin. Mo na iyang format, no? It's chromatin is basically DNA but in a different format. Ana, mo na bitog imong document din nahimo siyang PDF. It's the same document per lahi siya nga format. Or kato nga document himo nimong picture. Oh, it's the same document but again, it's in different format. So in this case, imong chromatin, the chromatin is the coiled version of your DNA, and it is the most apparent or most obviously seen in the S stage. But this is the kind of DNA or the format of DNA that is found in your nucleus or the nucleus of the cell that is in stasis or kanang wala nag cell division kana siya. But if mag-start na ng cell division, ang imuhang chromatin will hypercoil into a chromosome. And then that is found in your mitosis. Ayan. Ayan. All right. Thank you very much for your responses. And thank you very much for your answers. Now let us proceed to the stages of mitosis. So we're done with the preliminaries. We will talk about mitosis and after that, meiosis na dayon. So yeah, let's get it on. Okay. So mitosis again is a cell division of your body cells or your what do you call this, your somatic cells. And it is very important to learn more about this because we can understand how your body heals itself or how your body renews itself, no? Because again, all of the cells in your body except your sex cells are able to replicate through this way. So magpasalamat na sa mitosis kay once we get wounded, after like a while, after pila ka days, mobalik na siya sa normal. Or in that case, if deep kayo ang wound, you will develop a scar. Okay? Or for example, if you break your bone, ana lugar, naka minor fracture ka, your bone cells will start to replicate itself. But your bone cells will have a lower rate of mitosis versus your skin cells. That's why your bone cells, that's why when you have a fracture, it would take some time for it to fully heal, no? Like for example, when I fractured my tibia last 2018, was it 2018 or 2019? I think 2018. When I fractured my tibia, it took me I was out of commission or I did not work for 2 months. Kaya dili pwede nga lakaw-lakawan ako, no? Because it hurts, of course, it hurts. And ang imuhang bone cells during that time would start to go through mitosis, but again in a lower rate than that of your skin cells, and it takes some time for it to be fully healed. So mo na siya, no, na ay variations sa imuhang length of time in which your cells would undergo mitosis. So pasalamat ka sa mitosis ka, yay! We are healed. Charot. So, let's start with the M phase. No, yung mga M phase, the cell undergoes mitosis if it is a somatic cell, as I've said kanina. And if it is a sex cell, it will undergo meiosis. And it allows the cell to divide and produce new cells. So, ang imuhang mitosis actually is also a way for your or other organisms to reproduce asexually, asexual reproduction. Oh, because basically it has the same DNA, no? Ang iyahang diploid nga cell, it will just replicate into two diploid cells as well. Meiosis, on the other hand, from a diploid cell, from you, for example, from you, it will replicate into two. And remember what I said, kanang it will undergo the stages of cell division twice, no? So kaduha sad siya, so kanina yung duha ka diploid cell this will become 4 haploid daughter cells, no? So if that's the case, if it's haploid, it will need to unite with another cell. Mao nang imong egg cell o imong sperm cell. For you to become you, for you to become an organism, you will need a sex cell from your mother, biological mother, that is, and then another sex cell from your biological father. Father! All right. So, preparing for mitosis, of course, dara imuhang dara mag go in imong interface. Okay, remember that. So remember G1 checkpoint. In this G1 checkpoint, you are going to this checkpoint will be checking for the number of nutrients produced, the growth factors produced, and if there's DNA damage. If that's the case, if all of these are not met, dahil kini mong cell adto din siya sa G0, no? Dahil dara sa kaday, tulog sa kaday, ha? Resting state sa siya. Pero kung na-meet niya tanan requirements, ano siya, go, go, go! Laban lang! Ano siya? So, diretso rin ta sa imuhang S stage. Okay? Remember S stage? Dara mag-replicate imong DNA because we have been assured in the G1 checkpoint, yeah, there's no damage. Okay? Your DNA is fine. So you are free to replicate. So dara dayon mahitabo sa S stage ang replication. And then after the S stage, dana na dayon sa G2 or your GAP2, wherein in this particular checkpoint, it will check the cell size if it has increased, and of course, if correct ang DNA replication, na nahitabo dito sa S stage. And then of course, here in mitosis, you have your metaphase checkpoint, no? Tanawon nimo kung naka-attach ba ang spindle filament or spindle fiber sa imuhang kinetochores of your chromosomes. All right. I'll not talk about this anymore. Kasabot naman mo sa sa concept of your DNA having different formats. It's just that ang imong chromosome, okay? Your chromosome, which looks like this, no? Hypercoiled din siya. Hypercoiled version. Kini imong chromosome, di ba? Na-replicate naman imuhang DNA, no? So this is the original, this is the, di ba yung original, but this is copy number 1 and copy number 2, and each of this, kini siya, this is a chromatid, no? Now, since duwa mo sa kabuok, na-attach sila through a centromere, then right in the middle, therefore, since attach mo sila, kini silang duha, they are called sister chromatids. But as a whole, Jude, it's called chromosome. And as I've said kaganina, your spindle fibers will attach to these structures called your kinetochores that is found in your chromosome. And so that is the anatomy of your chromosome. Uning gawas na ang sunlight. Nawala ang dagom. Hey, look at the sun. Ito eh. And I will not talk about haploid and diploid cells anymore because you understand that concept, no? Diploid meaning you have your mama and your papa set of chromosomes. So 46 chromosomes all in all, 23 pairs. So 23 kapares, bale, if you're gonna count it individually, that's 46. And then when you say haploid, that is half of the chromosome. So 23 pieces chromosome. So one-sided. Okay. Now let's go to the phases of mitosis. So as I said, PMAT, prophase, metaphase, anaphase, and telophase. Actually, just looking at these pictures, no, makagets na mo what happens in these stages of mitosis. Let us start with your prophase. In prophase, the chromatin condenses into chromosomes. Remember, naka-coil siya dito sa kuan sa S phase, ang imong DNA naka-coil siya into chromatin format. So in prophase, mag-hypercoil pa jud siya into chromosomes. Ayan. On na. So kini siya mag-hypercoil ang chromatin, then mag-paris po dyan na sila, no? Ang paris, of course, each of these will be called sister chromatids, and giparis na sila because they are expressing the same gene and the same, the same allele, ana. So, that is your, chromosome, ana, mag-hypercoil na din siya. And then the nucleolus disintegrates. So kanang nucleolus kanang dense part sa imong nucleus, no? Kanang dense part nga medyo daghan kay chromatin dinha, and also dinha aside makita nimo ang production of your ribosomes. And then the mitotic spindle starts to form from your microtubules basically, no? Kining spindle is very important because it attaches to your kinetochores of the of the chromosome, no? Na siya mag-attach and siya din mag-pull later on sa imong chromosome. So that is your prophase. Now, in between your prophase and metaphase, you have this pro-metaphase, but actually this is still part of your metaphase, your prophase, no? It's just in between. So it begins after the nuclear envelope breaks down. So the nuclear envelope breaks down during the prophase, no? And the centrosomes move to the opposite poles, just like this one. And mag-ready na siya dinhi. Your centrosomes will be governing your your spindle fiber or your microtubules, no? Siya ang mag-control, siya mag-pull. Come here, come here, come here. And then naka-attach ning mga spindles sa kinetochores of your chromosomes. And then, the kinetochores found at the centromeres become apparent or mas obvious ang ilahang appearance. Kadto lang kay gapangatol ko. What's going on? Okay. All right. So let's go to metaphase. So sa metaphase, remember metaphase starts with an M. So M for middle. Okay? Why don't you just meet me in the middle? So dapat ang goal din sa metaphase is dapat align ang imuhang chromosomes right in the middle of the cell. Kay dara man siya mag-start o drag, no? Dara man siya mag-start o pull. And then the microtubules of the mitotic spindle attach and interact with the kinetochores of the chromosomes, as I said kanina, and the chromosomes align at the center of the cell in the metaphase plate. Okay, it's just an imaginary region. Right in the center of the cell. And then, naa diri imuhang M phase na imuhang checkpoint or M checkpoint, wherein it will check if these fibers right here, these microtubules, are really attached to your kinetochores. Because if they are not, then as I said kanina, nayusa ka cell nga gansi, nayusa nga cell nga nasubraan o chromosomes, no? So that's it. Now let's go to anaphase. Now, the letter that you have to remember here is your anaphase starts with the letter A, no? So in anaphase, A as in away, ana siya, going away from each other. Okay. So what happens here? The mitotic spindle pulls and separates the sister chromatids apart. That's the reason why they have to be aligned right in the middle, because they will be pulled apart by your spindles. And the chromatids, now called daughter chromatids, are then pulled towards the opposite poles. So away from each other. That's the key word there. And then you have your telophase, no? Remember the letter T? Ano siya? As in telephone. Bitaw joke lang. Okay, what happens in telophase? Daughter chromatids decondense. So, nag-separate naman sila na nasa sa opposite poles of the cell. So, kanang imuhang chromosomes, katong, di ba, half naman ang nabilin. Ang the other half kay naa sa pikas, no? Nasa dali. So, ano siya nga, hi, it's time to relax. Ano siya? So, iyang sort of like uncoil iyang self kay hypercoiled man to ganina, no, as a chromosome. So, iyang i-uncoil iyang self into a chromatid balik na mag-decondense siya. And then, mag-start to form imuhang nucleus. Hinahinay na siya ka-start, no? And then, in animals, the boundary of the new cells will be known as your cleavage furrow. So that this will be part of your cytokinesis. Mahina na siya. So in plants, on the other hand, cell plate siya. So cleavage furrow and then cell plate. And then after that, this lasts for like an hour. And after that, the cell will be, ano man, static na din siya. Mag G0 sa din siya. Chill lang sa siya. Chill. I'll just be here chilling, relaxing, ano siya. And depende na dayon kung mag-start na sad ang cell division. If that will start again, ang G0 nga cell, moadto na din siya sa G1. Okay, mabalik ka sa siya pagka G1. And your interface will last for about 24 hours or so, and then balik ka sa siya sa mitosis. So that's what happens. So mo na imuhang PMAT. So if you're going to look at here, kani siya, no? This hyperlapse ni siya nga cell division using the dyes and electron microscope, no? So this is really like cell division in motion. So you can actually see PMAP ana. Pero mas obvious jud kaayo ang imuhang kanang pag-align bitaw sa middle, and that is your metaphase. Very obvious. You can actually also see some of the phases of your mitosis dinhi sa kuan kini siya nga particular picture. And these are onion cells, no? And then here kini siya, this is mitosis but rapid lang ang produce. These are basically bacteria. Oo. So that is how they reproduce. Pero hindi na kapaspas ha. Rapid din siya or kuan din siya kanang hyperlapse na siya. It takes a while. It takes a while for your bacteria to reproduce itself. But it could take hours. It could take also days para inani kadaghan, no? But that's how they reproduce. They reproduce asexually through mitosis. I'll not talk about this anymore. Okay, let's check your understanding, Dolby. Identify the phase of mitosis that the following events belong to. Number 1, this begins after the nuclear envelope breaks down. Nag-break down imuhang nuclear envelope. Dito pa na sa kuan interface, no? So what happened? What begins after that? Yes, Juniel.
Prometaphase po, ma'am.
Prometaphase, all right. Any other answers? Any other? Hello? Nag-chat? Wala. Anybody? Nakatulog na mo?
Hi, ma'am.
Are you sleeping? Mm-hmm. Beto. Okay, that's prometaphase or pwede na sad siya, ano no? Actually, pwede rin siya, prometaphase. Prophase, actually. Or prophase. Nagbalik ka sa kadagom, uy. Dagom na sad sa amo, ah. Okay, number 2, the nucleolus reappears. Nag-reappear imong nucleus. So, after all that happened, nag-reappear siya. I'm back! Ano siya?
Telophase, ma'am.
Okay, correct. This is telophase. Telophase as in telephone. I'm just kidding. Okay, number 3. Lobat na daw akong tablet. Gadget lang. Okay, number 3. The nucleolus disappears. Appear, disappear, 1/2, 1/4, 1/5.
Prophase po, ma'am.
Prophase, ma'am.
Yes, that's
Prophase, ma'am.
prophase. Okay, correct. So prophase pa lang, ay mong nucleolus kay nag-disappear. I'm so sorry. Naglag siya kay nalobat na akong tablet. Okay, number 4. The chromosomes align in the middle of the cell.
Metaphase, ma'am.
Metaphase.
Metaphase.
Yes. And number 5, the sister chromatids move to opposite poles. Ihanggi, then iya din ana. Saan siya?
Anaphase, ma'am.
Anaphase po, ma'am.
Okay. Basta moving apart or pulling apart, anaphase. All right. Thank you very much for your answers. You have a firm grasp of the concepts, no? Okay, let's go to meiosis. Meiosis again is a cell division that is found in your sex cells or your gametes, no? Your sperm cells and your egg cells are produced through this kind of cell division. Now, the thing is, if you're going to differentiate between the egg cells and the sperm cells, no? For the women, kita ng mga babae, no? The number of egg cells that we will have throughout our lifetime has already been predetermined na dato pa lang sa naapata sa womb sa atong mothers, no? So, naanay mga polar cells din ha found in our ovaries, underdeveloped ovaries, and then igo na lang din na siya immature as we grow up, and then as once gani nga we have our menarche or your first menstruation, dara na dayon makaingon, dara na dayon nato mabalaan nga uy ang atuang egg cells that are found in our ovaries, they are ready for maturation. Pwede na sila, pwede na pwede na kaya but for the males, it's another story. For the males, you will have unlimited supply of your sperm cells, sana all. For the women, we go through menopause because again, menopause signals that we ran out of supply. Hurot na atong egg cell. But for the men, you can actually produce not egg cells, sorry, sperm cells until the day you die. And it's just that as you grow old, of course, mas minus ang production. Pero naagyapon. It's still there. And again, this is variable also. Okay? It depends from person to person. Of course, na ibang lalaki nga low ilang sperm count, das ibang lalaki nga bonggang-bongga. So it depends. But for the women, menopause. Okay. Let's go to meiotic division. So a meiotic division basically is the production of functional sex cells or gametes, and there will be a reduction in the chromosome number. Remember, from the haploid sa first phase kay haploid, diploid. From a diploid cell sa iyang first phase, no? So iyang first stage of division, kay diploid gyapon iyang kaka-produce, and then sa second stage haploid na dayon ang mga cells, no? So it's a reduction, there's a reduction in the chromosome number. So there will be two rounds, that's meiosis 1 and meiosis 2. But basically they're just PMAT, both of them PMAT. PMAT 1 as in prophase 1, prophase 2, prophase 1, metaphase 1, anaphase 1, etc. And your meiosis 2 would be PMAT 2. That's what the difference is. And your meiotic division allows restoration of original chromosome number during fertilization. So kailangan, if gusto magka-baby imuhang parents, kailangan magtagbo ang duha ka sex cells. Ayan. So if you're just going to look at this photo, no, it will, or this diagram, it will just give you an idea na of what meiosis 1 and meiosis 2 is all about. Actually, imong meiosis 1, I'll just give you an idea in a nutshell. In meiosis 1, naatay gitawag nga crossing over here in prophase. Crossing over. Okay. What is crossing over? Basically, ang duha ka chromatids, okay, ang duha ka not chromatids, not chromatids. Ang duha ka chromosomes, one from your father, one from your mother, mag-exchange na silang information. Okay? Because as we've established before, ang imuhang mag-abay ang duha ka chromosomes that are expressing the same gene, no? One from mother, one from father, mag-abay na sila. So they will both express eye color, hair color, nana, and then mag-exchange na silang information, no? So remember, kisa inyong father, si Chris Evans, kisa inyong mother, si Song Ye Kyo. So, usa dayon, pag abot dayon din tan-awon ninyo ho, na ay kanang lahi-lahi nga color, di ba? Na ay kanang murag variation. So in this case, there will be variations na usa makakuha og blue nga eyes, na usa makakuha og brown nga eyes, na usa makakuha og blonde ba si Chris Evans? Is he blonde? But we'll just use Captain America's face, okay? Blonde ang usa, then ang usa kay brown hair, etc. etc. Oh. So move forward din na siya through anaphase and telophase. So same, it's the same concept as before. So meiosis 2 is still PMAT. It's the same. Dili na dayon ang PMAT na imong makita sa imong mitosis, no? So walay etchos. Diretso na siya mitosis nga kanang pag-divide. So in this case, walay replication na mahitabo. There will be no DNA replication. Diretso na siya. So pag attach ni spindle fibers sa imuhang sa imong chromosome, half ra yung makuha jud, no? Half of the information. So from 46 mahimo siyang 23 na dayon. So makahimo na din siya daughter cells that are all haploid na sila. So mao na jud na siyang special features ni meiosis. Crossing over is really just one of those features really. So as I've said gidiri, ang imuhang sa imuhang prophase 1, you have your homologous pairs, no? What does homologous pairs mean? As in pare sa chromosome nga ga-express sa same gene. That's it. And then one is from Chris Evans, your father, and one is from your mother, Song Ye Kyo. Dayon mag-crossover sa magkuha na sila, ma-exchange. Then nana din kayo makita nga variations, no? As you can see there. No. One could come from like majority of the features would come from your father, and majority would come from your mother, and then asay recombinants, no? You call them recombinants, meaning kuan ana niya na ay kanang part ana niya nga wholly from the father and another part from the mother, etc. etc. So that is why na ay kanang mga times, no, na makita nimo na ay mga offspring nga pareho kay sa gitsura sa iyang mom or pareho kay sa gitsura sa iyang papa. Like for example, do you know Brooke Shields? Not Brooke Shields. I forgot the. Kailan ba Kaya Gerber? Kailan ba niya? Kaya Gerber. Basta model na siya. I-google na ninyo si Kaya. Amal kung sakto ako spelling siyang Kaya. O kaya ni Kaya Gerber. Her mother was a supermodel in the 90s. Actually, young mother is so beautiful. Way back when I was a child, I used to see her mother, supermodel, her mother endorsing L'Oreal. L'Oreal ba to siya or Revlon? Either of those brands. It's either L'Oreal or Revlon. And si Kaya is like a photocopy of her mother. Like that same face, exactly the same face. So that would mean it's a recombinant. It's not really dili man siya kanang exact to the photocopy, but recombinant siya pero ang kuan jud, majority anak niya would come from the mother. It would be like this one, kining pink. Pareho kasi namong, no? Same features, same expression. But if you see like offspring like kailan mo na Shiloh Pitt, anak ni Angelina o ni Brad Pitt, or si kanang anak bitaw ni anak ni Sunshine Cruz o ni Cesar Montano, kana siya, I forgot her name, I'm sorry. Kana siya, they are like kanang perfect kayang combination sa ilang mga nawong. Like if you've seen like Shiloh Pitt, ang iyang lips are from her mother, ang iyang hair is from her father, ang iyang blue eyes are from his father as well. Ang iyang nose, I don't know exactly where that nose comes from. Like iyang forma sa iyang nose, but maybe from her mother. And then perfect jud siya ang combination. Combination jud siya maklaro jud nimo nga kinsa ang parents because it's a combination. Hala oy unsa to na ako'y nakita ka ganina. Pacquiao ma'am sa papatanan ni liwat sad face. Bastos. Bastos ka rin, Jin. Okay.
Dili ako.
Liwat kaayo kay Angelina. Yes, but perfect si Shiloh, perfect siya ng combinations both mama and papa nga side, no? So true. So kana siya, it could be ang expression na niya would be the recombinants, no? Perfect na 50-50 ang iyahang pag-express sa iyahang chromosome. Okay. I don't talk about this anymore, ha? Kay kung naman i-repeat naman siya, no? Basta PMAT 1, PMAT 2. Oh. Ang importante na sa prophase 1, crossing over. That's it. And it will follow the same stages sa imuhang mitosis. PMAT gihapon. Alright. So, let us differentiate between the two, mitosis and meiosis, no? Ang imong mitosis, of course, ang iyahang it will start off as diploid cell and then divide into two diploid cells. Meiosis, on the other hand, it starts off as a diploid cell and then divide into two diploid cells, and then kay kaduha man siya mo undergo og kuan PMAT, so mo produce siya upat ka daughter cells. So here sa mitosis, two daughter cells ang iyang kaka-reproduce, and then identical siya, and then dinhi sa meiosis, it will reproduce 4 daughter cells but recombinants, no? Dili sila identical. All right. So sa paman ang difference nila, no? Kining mitosis, it happens throughout our lives, no? This happens until the day we die. On the other hand, kining koan, meiosis, this only happens during the reproductive years. Ayan. And then, your mitosis is involved in asexual reproduction. Your meiosis, on the other hand, is involved in sexual reproduction. So, in a nutshell, mauna ilahang difference. So, mitosis and meiosis, that's the difference. I want to talk about this, ha? Okay. Of course, why do we have to cross over? Nganong kailangan man na ay variation or na ay variances, no? Why is it that it is important that we don't look alike, therefore we don't have the same internal environment, we don't have with our siblings, no? Sa katong na mga igsuon, it is important na dili ta pareho og DNA. Naatay similarities, of course, because we come from the same parents, but dili jud siya 100% copy. Why? Why is it important? Because it facilitates genetic recombination, that's first. But why? Why? Because it enhances genetic variation, and as a result, this will increase our chances of survival. Now, why is that important? What are some examples of that? For example, kabaluman mo sa concept of dominance and recessiveness, right? Naay dominanting quality sa usa ka kuan, sa usa ka race, like, or sa usa ka species, like kita like for example kita lugar mga Asians, no? Unsay dominante sa to nga hair color? What do you think? Like all humans have this kind of hair color.
Black.
Black,
right?
brown.
Black, brown.
Black, brown, ma'am.
But for Caucasians, as a race, ang ilahang dominante nga color would be unsa man?
Blonde.
Blonde, yeah. Caucasians would have that. It's blonde. But as a whole, as a species, ang black hair is more dominant than that of your blonde hair, because black hair / brown hair is seen in more humans than that of your blonde hair. And sometimes, not sometimes, but most of the time, when you have a foreigner parent, a blondie father, a blondie mother, um, reproducing with a black haired parent, usually ang ma-express yun na will be either black or brown. Not so much with the blonde. Dili kaayo siya. Because it's recessive, no? Dili siya dominante. But what if simba ko palayo? What if you get to copulate with a very close relative? Like a brother, a brother or sister, yeah, or a cousin, kanang very, very close. So, since close kayo mo og DNA expression o close kayo mo og DNA qualities, that would mean that you would have the same dominant traits and recessive traits. Now, what if naamoy recessive trait nga, let's say for example, Down syndrome. Kanang Down syndrome, this is really a recessive trait. Recessive dyan siya. But what if kamong duha, ikaw, ang babae or whatever, naamoy Down syndrome duha, or recessive na siya. But nag-copulate mo, therefore, kining Down syndrome, this will become now a dominant trait. Dominante na siya. Nga naman, kay, mura ba ito siya positive, negative equals positive, and then negative, negative will become positive. Oh. So, inana siya, no? So, negative, negative, let's just think of it this way, negative, negative, or recessive, recessive, si Down syndrome, and then if i-combine mo ang DOCA recessive, this will become a dominant. No? May mo na siyang dominante. So, that would mean that if that's the case, you and your brother will copulate, no? Your child will definitely have Down syndrome. Ma-express na yun na siya. Na, hala, nalaki Down syndrome itong anak. Pero, well, wala man sa kuan. Wala man sa previous nato na makuan. Okay. Wala siya na-express sa uban. Wala siya na-express sa imong mga auntie, mga uncle, or even yung mga kanuno-nunuan because it's a recessive trait. Now, in this case, because you are copulating with a close relative, and then ang recessive trait kay na-express siya sa duha ka buo ka DNA from your mother and father, from you and your brother, therefore your offspring will definitely have Down syndrome. So, am I saying that children with Down syndrome have parents that are close relatives? Yes. Or not the parents, but pwede iyahang grandparents. Pwede na siya. Sir, yeah, na siya. So that's that's the case with kanang copulating with a close relative. There are some traits na dili kaaya-aya na dominante siya or ma-express siya. So, ang goal dyan aning crossing over is increases our chances of survival dyan. So, maminusan ang atuang pag-develop of diseases or pag-express sa diseases. Kay recessive man na sila. So, am I saying that it's better to marry a foreigner? Somewhat. Because,
Yes, daw na sa phone.
charot kisa din Jin. Uy, check up kisa din Jin. Somewhat. Why? Because kita magud like kita mga as a race, no? Kita mga Asians, and then the Caucasians and then the others kana sila. We have kanang similar kanang propensity for diseases. Like for example, kita mga Pilipino, there's a huge possibility of us developing like cardiac diseases. And then kini din mga Caucasians, there is a higher possibility of them getting neurogenic na mga diseases. Kana bitong multiple sclerosis, ALS, kana siya na Caucasian kay na siya nga sakit. It's very inherent to them. So, for you to increase your chances na inyong offspring will not develop these diseases or not express these diseases, pagka mo, ikaw, si Song Ye Kyo, makikombine with Chris Evans, pak, na, you have amazing genetic variation. Ana. So, definitely you won't have, not definitely, but there's a huge chance that you don't have the same recessive traits you don't have the same dominant train. So there is really like difference. Afam is the kikuno daw. Ambot ni mo rin, Jin. Ikaw bahala, anahan. Hindi ako sinabi. Hindi ako nagsabi niyan. Charot. Okay, so that is the importance of genetic variation, Junasha. Okay, so last check-up na lang ni. Identify the term being described in each statement. Number one, this event refers to the exchange of segments of the homologous chromosomes during meiosis 1.
Crossing over, ma'am.
Yes, crossing
Crossing over, ma'am.
And exchange of segments. Ayan. Number two, this structure guides the chromosomes in migration during meiosis. Unsa man to siya? Kato bitong mga wire.
Metaphase, ma'am.
Ha, dili, structure.
Eye structure. Koan.
Na siya, naka-attach siya sa kinetic cores of your chromosomes. Unsa man na siya? Structure. Na, kato siya. Sito gani. Kita mo sa drawing? Dili siya face. It's a structure. Structure. Microtubules, yes. Okay, pwede siya. Microtubules. Pwede siya spindle, mitotic spindle. Pwede na siya. Okay. And lastly, we have, this is the area in the middle of the cell where chromosomes align before segregation. What do you call that? Katungtunga. Chuk. Na siya? What do you call that? The one, why don't you just meet me in the middle?
Metaphase, ma'am.
Metaphase what? Metaphase?
Metaphase 2, Charles.
Metaphase plate.
Area man. This is the area.
Plate, ma'am. Metaphase
Plate.
plate.
Ayan. Metaphase plate.
Plate.
Ayan.
Ay, plate.
So last na lang to the concept. Very, very last. I'm not talking about this in Merck. Kabalo naman mo, ani. Kaya siya. Okay. That's a concept. Okay. What your cells will not stop replicating? What if your cells will not stop mitosis? How do your cells even know when to stop? Okay. Kaya di ba, kabalo naman ta sa regulators on kanus mag-start ang imuhang cell division, katong imong proteins, right? Internal and internal regulators, just like your protein kinase and your cyclins. And then you have the external regulators, your growth hormone. But what if, what makes the division stop? Okay. So nato gitawag na density, ang imong cells na siya density dependent inhibition. Ang pasabutan niya kay imong cells makadetect actually kung puno na ang space, no? If there is no more space for new cells, it will not go through mitosis anymore. So nana sila detectors ana. That could be one. Another one would be the Hayflick limit. So it has been discovered by Hayflick that a cell, a single cell, a single somatic cell can actually only go through mitosis up to 50 times. Only up to 50 times. And beyond that, wala na dayon. That's the maximum. But it depends, no? As we age, of course, muhinay na siya. So, mura na iyang limit. So, pwede na siya. Stop. But what if your cell does not stop dividing? What if di siya makastop, no? Dili siya makaregulate og tarong. That's what you call as cancer. Ayan. That's cancer basically. I know you know what cancer is, right? So cancer is, you know what cancer, you've heard of cancer. It's all around you. A lot of people have this. So hopefully wala ta, no? Kita tanan, wala ta di kini nga classroom, wala ta inana. Cancer basically is your inability of your cell to stop mitosis or stop replicating. But the thing about cancer is, ma'am, di ba okay naman, ma'am? Okay, it's just a healthy cell. The thing about the cancer cell is it is unhealthy. Ano, unhealthy man siya. Because wala man siya natarong o go through mitosis. Meaning, dili sakto iyang pag-replicate sa DNA, so there will be like a lot of mutations happening there. Or it could be that na-arrest ang iyahang PMAP, arrested development. Yes. Na ay arrested part sa imong PMAT. It could be prophase, metaphase, anaphase, telophase, any of that. Any of those phases. So, na-arrest or na-stop siya, dahil nagkalit lang niya siya, shoo, nikalit lang niya siya cytokinesis. Wala pa gay nahuman, o pag nahuman ng metaphase pack, nag-ano na din siya. Pwede na siya, no? Now, when cancer is very rapidly producing, dali ra kay siya makaproduce of more cells. And these cells, they will produce veins and arteries that will supply, that will give them a blood supply. Maunang kanang manghina jud na inyong mga cancer patients because a lot of their blood supply will go to the cancerous tumor. Dili dadto sa imong mga healthy cells mo nang magniwanga jud na sila, no? Pero gakadako ilahang tumor. So that's the sad part about it. So how can it be managed? How can it be destroyed? Through radiation, pwede. Because radiation can actually kill rapidly dividing cells. However, it is non-differentiating, meaning dili siya mopili kung cancer cell ba na siya or hair cell. Kaya di ba ang hair kay dali man sad na siya mo reproduce, or skin cell. Mo nang yung mga cancer patients after radiation therapy, they don't feel well, no? Their skin is very dry, their hair is very thin, ana. So those are the effects of it. Pwede sad siya chemotherapy na diretso niya gina-attack ang cell through the DNA. And that is, again, ang ihasang target na is rapidly dividing cells but non-differentiating. Di po siya differentiate between cancer and healthy cells. So munang mag-chemotherapy na sila pero ma-weaken sila. Mag-nausea, so, magsukaha din sa after, or they don't feel well right after. So that's the sad part about it. Ayan. So that is it, basically, on our lesson. No? Are there any questions? But if not, any questions, it's already 9:59. I'm so sorry. I took up a lot of your time. But if you have questions, kindly reserve your questions tomorrow. We will meet tomorrow for our next topic on, what's our next topic? Cell transport tomorrow. So reserve your questions and I'll be answering them tomorrow. No? So for now, exit sa tanong, dili lang sa tataman because it's already 10 o'clock. I have a class at 10 o'clock pa. And we will continue on with our topic on cell transport tomorrow. So STEM 4, thank you very much for coming to our synchronous class this morning. I'll see you again tomorrow. Please be prepared. I will also publish your second short quiz right after our class. I free na hapon because I have straight classes today. And I will also publish the link to our recording. Okay? So thank you very much and I'll see you again tomorrow. Goodbye, STEM 4.